A good day to one and all listening into this presentation. My topic for discussion is 2-deoxy-D-glucose and its role in COVID-19. I'm Dr. Hemant Vasan, a final year resident in the Department of Radiation Oncology. This topic was given to me by my guide and professor and HOD, Dr. V. Lokesh, who has asked me to delve a little deeper into this molecule. Mm, Contrary to the popular notion, 2-deoxy-D-glucose is not a new molecule. Um, it has been around for quite some time. In fact, it was developed and modified in early 1900s. What exactly is 2-deoxy-glucose? 2-deoxy-glucose is a decoy of the actual glucose molecule, where the glucose molecule is tweaked and the second hydroxyl group is replaced by hydrogen. Now, what does it do? In the early 1900s, it was shown that this molecule inhibited the fermentation of glucose by yeast cells and confirmed that this glucose analog inhibits glycolysis of glucose in a variety of normal and tumor tissues as well. Now, central to this discussion, laying the foundation for further slides, we'll have to get into what exactly 2DG does. It blocks the glycolytic pathway. What is the glycolytic pathway? The glycolytic pathway is a central metabolic pathway which is characterized by the breakdown of the glucose molecule and its metabolism into pyruvate for the generation of ATP, which basically fuels the cell and all the cellular processes. So 2DG, it blocks two key enzymes catalyzing glycolysis. It non-competitively blocks hexokinase and it competitively blocks phosphoglucose isomerase. And these processes as well were proved to be inhibited by 2DG early in the 1900s. So this is to reiterate the fact that 2-deoxy-glucose is not really a new molecule. It's something that was developed very early in the 1900s, very thoroughly researched, used in various experiments and research activities for diagnostic as well as therapeutic purposes. That being said, we'll move on to one of the most fundamental processes that would lay the foundation to understand the role of 2DG in the COVID-19 setting. So I'm going to be talking to you about the Warburg effect. Now, what is the Warburg effect? The Warburg effect was fundamentally observed and described in cancer cells, and it was designated as a hallmark of cancer. So what exactly does this effect entail? It is a very peculiar phenomenon in the glycolytic pathway characterized by a paradoxical shift of the metabolism of pyruvate predominantly to lactate instead of oxidative phosphorylation. And this is despite the presence of adequate oxygen. Hence, this is known as aerobic glycolysis. So let's look into the diagram. So when you have well-differentiated cells, your central metabolic pathway, that is glucose metabolism via glycolysis, in order to generate ATP, occurs via oxidative phosphorylation. So you have your glucose that's converted to pyruvate, that's taken into the TCA cycle, and oxidative phosphorylation in the presence of oxygen. although some of it may be converted into lactate. This is a very high yielding process. It produces about 36 ATPs for a molecule of glucose. And in the absence of oxygen, glucose is converted into pyruvate, and pyruvate is converted into lactate. And this process generates only 2 ATPs. Now, what happens in the cancer cells? What was described in the cancer cells? It was seen that despite the presence of oxygen, there was a metabolic switch of the glycolytic pathway. Glucose was converted to pyruvate, but majority of this pyruvate was converted into lactate, which was a very predominant finding in these cancer cells and very key metabolic process that was observed. Now, developing from Otto Warburg's data from the 1920s, it has become common knowledge that increased glucose metabolism is a universal principle of cancer cells. And the key mechanisms at play which sustain this process involve the following: rapid ATP synthesis via increased production of hypoxia-inducible factor alpha, which increases glucose uptake by upregulating glucose transporters, the expression of glucose transporters. There is increased glucose phosphorylation, upregulating hexokinase 2 expression. We saw that hexokinase was a key enzyme in the glycolytic pathway. It stimulates the switch to aerobic glycolysis by facilitating the inhibition of oxidative phosphorylation. So what this Warburg effect does is it creates a state of glucose hunger. And you have every possible mechanism to uptake the glucose and process the glucose. It also upregulates cellular biosynthesis via producing the building blocks, that is the nucleotides and the carbon moieties, which are supplied by branching pathways or intermediate metabolic pathways of the glycolytic pathway. These include the pentose phosphate pathway, the serine pathway, and glutamine metabolism. The next mechanism is a very key mechanism that will repeat itself throughout this presentation. So there is a phenomenon which is known as glycosylation. So what exactly is this post-translational glycosylation? Here, newly translated proteins, which are, I would say, immature, they go through the process of glycosylation in the endoplasmic reticulum. They go through post-translational modifications, which are complex modifications for building up the structural as well as the functional integrity of the newly translated protein. And one of the key mechanisms at play for this is glycosylation. So, Warburg effect maintains the endoplasmic reticular homeostasis through proper post-translational glycosylation of key cellular proteins. What else does it do? Now, as we saw in the previous slide, there is a majority of the pyruvate being converted into lactate. And we know that lactate acidifies its environment, which enhances invasion and depresses local immunity. So these are the key mechanisms at play which sustain this Warburg's effect in cancer cells. How does 2-DG come into play? So because of its similarity to glucose, administration of 2-DG would block this glycolytic pathway altogether, block the production of ATP because these tumor cells depend on glucose as their main energy source, and it essentially starves the cell to death. It depletes the intracellular ATP, The low ATP induces energy stress, which activates various protein kinases and the mTOR, and this stops cap-dependent protein synthesis and results in growth arrest. What else does it do? Now, I spoke to you about glycosylation, which is a key process in the maintaining the structural and functional integrity of a newly translated protein, which occurs in the endoplasmic reticulum. So, this 2DG, apart from blocking glycolysis itself, it blocks glycosylation. And how does it do this? It is a decoy to it's a decoy to mannose, so which is an important protein in glycosylation. So chemically, it is also 2-deoxymannose, and it competes with the mannose in growing lipid-linked oligosaccharide chain during the initial steps of N-linked glycosylation. And this mannose-like property of 2DG results in misfolded proteins, leading to endoplasmic reticulum stress-induced cell death. 2DG also plays a role in inhibiting angiogenesis and metastasis. This flowchart just summarizes all the effects of 2DG in the context of the Warburg effect and glycosylation. So it blocks hexokinase, PGI, also acts as a decoy to mannose, blocking glycosylation and ER homeostasis, blocking glycolysis and energy metabolism, blocking intermediate metabolic pathways, which are crucial to providing the building blocks and nucleotides for biosynthetic processes and synthesis of DNA and cell cycle proliferation. So that was 2DG and the context of its effects as an anti-cancer agent. Now let's come to 2DG versus viruses. Now I have documented here chronologically various evidences to show that 2DG has activity against viruses via different mechanisms, albeit, and against various viruses. So we are establishing the fact that 2DG is effective as an antiviral through various mechanisms. Now, if you carefully look at many of these mechanisms that it interferes with through the ages, a lot of them involve glycosylation, the glycosylation process, or alteration or disruption of glycoproteins, which are essential to viral replication. However, you can see another mechanism prevail in context of the human papillomavirus, where it directly acts on the expression of oncogenic genes, that is the E6 and E7 gene, which we know have been proved to be notorious to produce and facilitate carcinoma of the cervix. Here we're showing that 2DG accumulates in viral infected cells. And in this instance, apart from disrupting glycosylation and causing an unfolded protein response leading to misfolded proteins and eventually truncated proteins which are dysfunctional, in the instance with rhinovirus, which was recently observed, we can see that it shifts the carbon flux to a catabolic process away from production of important nucleotides and macromolecule production, which are essential for viral replication. So here are several instances to show that 2DG has potentially a lot of antiviral properties. Again, we come back to the Warburg effect, which serves as the common denominator. This phenomenon of the Warburg effect is not unique to only cancer cells. It is seen in rapidly proliferated cells, and It is also seen in virally infected cells. So, metabolic phenotypes conferred by virus infection often mirror metabolic changes seen in cancer cells, such as upregulation of nutrient consumption and anabolism to support viral replication or rapid cell growth, respectively. For example, cancer cells and virus infected cells commonly exhibit the Warburg effect, increased glycolytic metabolism in the presence of adequate oxygen for oxidative phosphorylation to supply reducing equivalents and precursors of macromolecule biosynthesis. Increased nucleotide and lipid biosynthesis are two other metabolic alterations associated with tumorigenesis and rapid cellular proliferation that are also seen in various viral infections. In theory, the inhibitory mechanism of 2DG could also be applicable to other viruses, which we saw. And 2DG has been found to have wide-reaching effects on glucose metabolism in tumor cells under hypoxic and normoxic conditions, as well as in endothelial cells and viruses. Now, coming particularly to the SARS-CoV-2 virus and COVID-19 infection, it is now common knowledge that diabetics and people with metabolic syndrome, obesity, uncontrolled blood glucose levels are at greater risk to develop the severe form of COVID-19. This does establish a link between deranged or elevated glucose, deranged glucose metabolism in diabetes, and SARS-CoV-2 infection. Now, this is a brief flowchart which shows the same. You have SARS-CoV-2 infection, which through various mechanisms elevated inflammatory cytokines, interferons, increase in reactive oxygen species and interleukins, derangement of the renin-angiotensin-aldosterone system, and elevation of fibrinogen and D-dimer lead through various processes, and hypoglycemia being central to most of these damaging processes, causing acute lung damage, vascular endothelial damage, increased blood viscosity, which eventually lead to cardiovascular events, thromboembolism and DIC, leading to cardiovascular and all-cause mortality increase. A very nice analogy was explained to me by my professor and HOD, Dr. V. Lokesh, and he compared this infection and inflammatory process that ensued with SARS-CoV-2 with a forest fire or a bushfire, which literally consumes everything in its path and it sweeps through, leaving everything damaged behind it and going ahead to damage everything ahead of it. And this is what happens in the lungs of, and eventually in the systems of people who are infected with the SARS-CoV-2 and who develop a cytokine storm, which propagates, which is a sort of a self-propagating event, like these bushfires. Again, going to the increase in morbidity and mortality, attributing glucotoxicity and infection of SARS-CoV-2. So there is dysregulation of glucose metabolism leading to endothelial damage and leading to tissue hypoxia, interstitial lung damage, increasing thromboembolic risk, which ultimately leads to all the classical finalities of SARS-CoV infection, that is acute respiratory distress syndrome, deep vein thrombosis, and multi-organ dysfunction. Adding to this, Kodo et al. In 2020 showed that elevated glucose levels enhance SARS-CoV-2 replication and cytokine expression in monocytes. SARS-CoV-2 infection induces glycolysis in human monocytes. Glycolysis is necessary for SARS-CoV-2 replication and monocyte response as well. The mTOR hypoxia inducible factor 1A axis is necessary for SARS-CoV-2 replication. Mitochondrial reactive oxygen species mediated hypoxia inducible factor 1A stabilization sustains SARS-CoV-2 replication and cytokine production in monocytes. SARS-CoV-2 induced metabolic reprogramming of monocytes directly affects T cell response and lung epithelial cell death. So here we're seeing multiple mechanisms by which uh, he has established a link between elevated glucose levels, SARS-CoV-2, and its um, importance in replication and response of a cell infected by SARS-CoV-2. I'm sure a lot of you or many of us would have seen these images, which were made very popular by uh, media, social media, television, newspapers, which are these plaque images of the 2-DG drug that has been recently introduced as a new ammunition towards fighting against COVID-19. Although there is not much published data, these were some images that were published in the Press Information Bureau. So essentially, what this shows is you have these plaques, which are viral plaques or sort of colonies of infected cells, as you can see, which are revealed as these white spots on these purple background plates, which they have labeled as SARS-CoV-2 infection. And these serve as controls to these petri dishes, which show the SARS-CoV-2 infection in the presence of 2-DG. And visually, it's striking because you can see a far fewer number of plaques as compared to the control. And this statement, which was made in the clinical trial document, that is, it was chosen based, this drug was chosen to proceed with clinical trials based on its in vitro inhibition potential. And these studies were carried out by the Institute of Nuclear Medicine, INMAS, and the DRDO, which have launched this new drug. Now let's summarize the actions of 2-DG in a SARS-CoV-2 infection and its potential target mechanisms. We'll start with blocking virus attachment, replication, and assembly. So a recent study, which was actually done and published in March of 2020, virtually through simulations have shown that the structure of 2-DG fits into several of the binding sites that allow COVID-19 to attach as well as to replicate once it gets into the cell, potentially targeting viral attachment and replication. So the authors of this paper show that docking of 2-DG with the main protease 3CL-Pro and endoribonuclease of COVID-19 is significantly better than that of standard antiviral drugs, lopinavir and favipiravir. Each of these sites is required for proper viral replication and infectivity. So we'll go on to an image. So we are talking about blocking viral attachments. You can see the classical coronavirus formed by the corona or a ring crown of its spike glycoproteins, which attach to the ACE2 inhibitors. So you have the mechanism which blocks attachment, and then you have mechanism which blocks the proteases. So the proteases act over here, which breaks down the newly formed, newly translated viral proteins in the cell. Next, you have, it has been shown by inhibition of glycolysis by 2DG, as well as by inhibition of glycolytic enzymes, that it abolishes viral replication and cytokine response, placing glycolysis as a key upstream event during SARS-CoV-2 pathogenesis. Although the critical glycolytic intermediate final products mediating such effects was not determined. So, SAR-2DG is affecting glycolysis as well as glycosylation. Now, its effect on glycosylation would be important over here when you don't have perfectly glycosylated proteins, such as the spike and other viral glycoproteins, which are altered, then it blocks potentially immune evasion, because these spike glycoproteins and other viral glycoproteins, they are speculated to be involved in immune evasion by shielding and shedding. So these spike glycoproteins, which are studied around the virus, they have been speculated to have properties of evading the immune system by some sort of a camouflage mechanism that is being researched. Also, many of these viruses shed these glycoproteins once they're out to avoid detection. That is also a theorized property. So, when glycosylation is blocked, there are improper post-translational modifications that occur, which blocks these immune responses. Then blocking the hyperimmune response. So the MT-ROS and HIF1A is necessary for CoV2 replication and monocyte cytokine production, and monocyte-derived cytokines drive T cell dysfunction and epithelial cell death. So this is a mechanism that can be blocked by 2DG. Also, pro-inflammatory innate, that is via the neutrophils, and the M1 pro-inflammatory macrophages, as well as adaptive immune responses via cytotoxic lymphocytes, B lymphocytes, and cytokines, and consequently their downstream cytokine production. These, they thrive on aerobic glycolysis. So we are coming back to our foundation, which is Warburg effect, which is nothing but aerobic glycolysis. And these can potentially be attenuated by 2DG effects. Now, another thing that happens, which may not be central to the virus as such, but these COVID-19 infections and pneumonias, they cause hypoventilation of the affected regions due to the pneumonitis, which causes alveolar hypoxia. And this hypoxia induces aerobic glycolysis in the endothelial cells and platelets. So this in turn activates and sustains vasoconstriction and platelet microthrombosis, which we saw in the earlier slides, that lead to those cascading damaging effects of thrombosis and pulmonary hypertension, resulting in extensive lung damage. And these processes, because they're sustained on aerobic glycolysis, they can be potentially an area of target by 2DG. So that was about how SARS-CoV-2 infections can be thwarted by the effects of 2DG, potentially. Some are theoretically proven, some are still in study, and the exact mechanisms remain unknown, although we do have the drug in phase 3 clinical trials and about to be launched right now. Again, this is a slide to summarize the processes that occur with SARS-CoV-2 infection. There is altered glucose metabolism, increased pro-inflammatory cytokines leading to T cell dysfunction and epithelial cell death, and increased replication of the virus as a result of these processes. And these are potential areas of target by 2DG. Now let's come to another link, that is 2DG and radiation therapy. Now, this was actually also studied, interestingly, much earlier in Kidwai Memorial Institute of Oncology in my institute. Way back, it was studied in gliomas or brain tumors. And this is where 2DG was used as an anti-cancer agent along with radiation therapy. So let's look into this. Glucose usage in transformed cells and tumors is increased, and tumor cells derive a large part of their metabolic energy from the glycolytic pathway that we've already seen. It was predicted that inhibitors of glucose transport and glycolysis could differentially inhibit repair processes within these cells, leading to an enhancement of the radiation damage. So this was the proposed mechanism. Subsequent studies demonstrated that the presence of 2DG, an inhibitor of glucose transport and glycolysis, during the first few hours of radiation, could indeed inhibit repair of DNA lesions and potentially lethal damage, thereby enhancing the radiation damage in various cellular systems with high rates of glycolysis like cancer cells, under normoxic or euoxic, as well as hypoxic conditions. And interestingly, under similar condition, a decrease in radiation damage has been observed in normal cells. Therefore, combining ionizing radiation with 2DG provides a unique opportunity to selectively destroy tumors by differentially enhancing the radiation damage in cancer cells and at the same time preventing radiation injuries to normal tissues. The multiple mechanisms underlying the sensitization by 2DG have been annotated. There is depletion of energy, disturbed redox balance, and altered end-linked glycosylation, as we've previously seen, leading to the unfolded protein response, inhibition of DNA repair, impaired cell cycle regulation, altered calcium influx, and apoptosis. It is pertinent to note that the radiosensitization of tumors in mice by 2DG is partly attributed to the immune stimulatory effects by combination of 2DG and radiation involving the restoration of CD4 and CD8 ratio, shift from Th2 to Th1, reduce IL-17, improved antigen presentation, enhanced NK cells, macrophage repolarization, as well as functional stimulation of phagocytic activity and decrease in immunosuppressive network. Besides this immune effects, 2DG with radiation generates anti-inflammatory responses. It mitigates bacterial infections. These studies have also shown that 2DG alone could enhance antigen presenting ability and functionality of macrophages and reduce tumor necrosis factor alpha while enhancing interferons. So we have seen that 2DG has a key role to play alongside with radiation. Now let's come to a very interesting topic, and that's being explored worldwide. That is the use of low dose radiation. What is this low dose radiation and hormesis? Hormesis is a terminology that, to be honest, even I've come across recently. So it's defined as a biphasic dose response to an environmental agent characterized by a low dose stimulation or beneficial effect and a high dose inhibitory or toxic effect. So essentially what it says is you have any environmental agent, or you could say a drug, or in this case we talk about low dose, and low doses would stimulate or have a beneficial effect, and high doses would have an inhibitory or a toxic effect. And this is termed as a hormesis. And this hormetic immune response is the crux of the mechanism of action of low dose RT. Now, the exact mechanism is elusive. It is a highly complex process. And this hormetic response was elucidated via various murine mouse models and in vitro cell models. And as we spoke about in the previous slide about the functionality of macrophages and the repolarization of phagocytic activity and macrophage repolarization. This macrophage repolarization seems to be the hormetic response that low-dose RT confers. And this is also a very central process of research. So what exactly is this macrophage repolarization? So you have these two phenotypes of macrophages. That is the M1 phenotype, which is pro-inflammatory, and the M2 phenotype, which is anti-inflammatory. And what is theorized and elucidated in these models is that low-dose RT, that is less than a gray, would enhance the M2 switch to the M2 phenotype of these macrophages, exerting a pro-inflammatory effect. Sorry, an anti-inflammatory effect, which causes tissue repair, matrix remolding, immunosuppression, and angiogenesis, as opposed to the pro-inflammatory phenotype. When you put all this together, you can see a potential new ammunition in the war against COVID. That is to combine low-dose RT and 2DG as a potential effective therapeutic strategy to combat COVID. And this is also an area that's being explored apart from the plethora of other available strategies, which include supportive care, biologics, natural compounds, pharmacological agents, and 2-deoxy-glucose alone itself, or its pro-drug versions which are being studied. This is a potential area of exploration to combine the hormetic effects of low-dose radiation and the potential multitude of targets that 2-deoxy-glucose has, and their synergistic response in cases of moderate to severe COVID-19, where we can tackle both the infectious component that would be attributed to 2DG and the inflammatory component that would be attributed to the low dose radiation predominantly. So with that, I would conclude this discussion, and I hope that this was a good overview, and this would dispel the notion that 2DG is a new molecule. It is not a new molecule. It is a molecule that has been around, which is being repurposed and further researched into to battle against the ongoing pandemic. Thank you.